Hello, this is my video on blood-brain barrier biochemistry and drug delivery challenges. So starting off, we're going to talk about what the blood-brain barrier is. The blood-brain barrier is a semi-permeable membrane that encases the cerebral blood vessels. And the purpose of this membrane is to regulate the movement of ions and other molecules between the brain and the rest of the body. This barrier is made of endothelial cells, the capillary basement membrane, and astrocyte end feet. These different tissues work together to keep toxins away from the brain and to keep nutrients from getting flushed away out of the brain and into the bloodstream. Diving more into the purpose for each structure, we see that the endothelial cells form these tight junctions using transmembrane proteins, and they pull the endothelial cells, the membranes of the endothelial cells close together, which blocks the passive transfusion of like larger and more polar solutes into the brain. Those tight junctions really only allow water-soluble molecules that are smaller than about 150 Daltons to pass through. Following that, we have the astrocyte end feet, and they make up a sheath of cells that encase the vessel and kind of just aid in the tight junctions and the mechanism of everything. Then we have the endothelial cells, which are kind of like the meat and potato of the blood-brain barrier. They create a barrier around the blood vessel that limits the transfusion of certain molecules using those tight junctions. And these are, of course, the most important parts of the blood-brain barrier because they are the main factor or the main defense in limiting or allowing for the transportation of molecules in and out of the brain. So in order to understand what kinds of molecules can pass through the blood-brain barrier, we must first understand the ways that a molecule can pass through. Generally, there are 4 different pathways that a molecule can take to pass through the blood-brain barrier, the first of which being paracellular diffusion. This is where the molecules must pass through the tight junctions between endothelial cells. This greatly restricts the passage of like larger and more charged molecules because the passage is so small due to those tight junctions. Generally, as I mentioned before, only water-soluble molecules that are less than 150 Dalton in size can just pass through there. The next pathway is through transcellular diffusion, which is another form of passive blood-brain barrier transportation. However, in this example, molecules must be able to diffuse through the endothelial cells. So this mechanism usually demands that molecules must be both lipid soluble and less than 600 Daltons in mass. Following that, we have absorptive transcytosis, which allows for larger positively charged molecules to pass through the membrane. The mechanism of this is a positively charged molecule will bind to the negatively charged membrane of the endothelial cell. This molecule is then invaginated by the peripheral endothelial phospholipids. The newly formed vesicle then will move to the other side of the cell, will bind to the membrane again and release its contents into the brain or the central nervous system. And then finally, we have receptor-mediated transcytosis, which requires a ligand to bind to a surface receptor on the endothelial cell. This then induces a conformational change where the receptor ligand complex internalizes into a vesicle that will travel through the cell and then be released onto the other side. This pathway allows for very specific delivery of molecules into the brain because these molecules must first bind to the specific receptor that brings them through. So now that we understand how molecules can get through the blood-brain barrier, here are a few examples of drugs that use those pathways. The first examples are molecules that can just easily diffuse through the membrane. Some examples are alcohol and anesthetics. Alcohol is water soluble and also very small, therefore it can easily permeate through the paracellular space of the endothelial cells. It'll just slide between the in between the tight junction of the endothelial cell and enter into the brain. Following anesthetics, they're lipid soluble, so they can just diffuse through the membrane pretty easily via lipophilic transcellular transportation. Some examples of absorptive transcytosis are penetratin, and TAT. Um, these are cell-penetrating peptides, so they can cross the membrane via absorptive transcytosis. They're cationic and kind of in like a truck and trailer method. They can be linked to a drug, in attempt to pull that drug in through and across the blood-brain barrier. In one study that I found that TAT and another cell-penetrating peptide QLPVM were used to transfer doxorubicin, which is a cancer-fighting drug that inhibits topoisomerase II, as well as um, erlotinib, which is a growth factor inhibitor for tumor cells. Um, and they use the QLPVM and the TAT in order to transport those molecules into the brain to fight glioblastomas. And so they did this by forming a membrane around the doxorubicin and erlo, and then attaching transferrin and the cell-penetrating peptides to the outside. Um, this allowed for the cancer-fighting drugs, doxorubicin and erlo, to be able to be pulled through the membrane and enter into the brain where they can be used to fight against brain tumors. Um, Finally, an example of a drug that uses receptor-mediated transcytosis is insulin. Insulin binds to the insulin transporter in the blood, which then invaginates the receptor ligand complex and transports across the blood-brain barrier to be released.